Hello, dear colleagues. My name is Valeria Lobanova, and I would like to introduce you to our work about amino acid shifts in the mitochondrial genome during the evolutionary path. Mitochondrial DNA is circular and it is replicated asynchronously. One chain, which is called a heavy chain, stays single-stranded during replication, and it causes mutations on the heavy chain. The most common transitions is C to T and A to G. When we see increased frequency of C to T mutations on the heavy chain, we expect that we will see an increasing frequency of G to A on the light chain, and vice versa. C to T on the light chain should cause G to A on the heavy chain. So, in case of the same mutations rate on both chains, we expect that the number of C to T will be equivalent to G to A on the heavy chain. But from previous work, we saw that the number of C to T is much higher than G to A. It means that C to T mutations happen predominantly on the heavy chain. The same logic works with A to G mutations and vice versa, T to C. In the end, C to T mutations will be more frequent than T to C, and this is asymmetrical mutagenesis, which can cause nucleotide content shift. But what about an amino acid shift? It has been shown before that an unbalanced change in the amino acid composition of the encoded proteins exists in the long-term evolution of the genomes of living organisms. Thus, the frequency of some amino acids increased, it's called gainers, or decreased, it's called losers. However, it is unknown what evolutionary driving forces determine this trend. Here, we hypothesize that the mitochondrial genome with strongly asymmetrical mutational spectrum, excess of G to A and T to C on the light chain, or equivalent to them A to G and C to T on the heavy chain, may determine gainers and losers among amino acids in the evolutionary process of living organisms. If we are right, we can predict the amino acids gainers and losers based on asymmetrical mutational spectrum rules. All genes except ND6 of human mtDNA in all databases are written as a light chain. So, if there is C to T on the heavy chain, we read it as G to A on the light chain. The same situation with A to G on a heavy chain, we read it as T to C on a light chain. When the first or second base changes according to the mutational spectrum, we expect that proline, histidine, glutamine, asparagine, lysine, and threonine will be gainers, and lysine TT, phenylalanine, cysteine, tryptophan, glycine, and valine will be losers. This is our prediction, and further, we had to check it. This work is focused on testing the prediction on eukaryotic and bacterial data. We used the protein BLAST to find orthologous mtDNA genes among alpha proteobacteria, fungi, plants, invertebrates, and 5 classes of vertebrates: breathing fish, amphibians, reptiles, mammals, and birds. We analyzed only organisms with all 10 genes present. ATP8 and MD4L genes had fewer orthologs among bacteria and eukaryota, and in the 6th gene, considered separately because it's the only gene coded on the other chain. We compared the frequency of gainers and losers in these genes and got a steady trend towards an increase in the number of gainers and a decrease in the number of losers, according to evolution of mitochondria. Alpha proteobacteria were taken as outgroup because it is the closest existing relative of protomitochondria. We can see that the number of gainers in vertebrates much higher than in invertebrates, fungi plants, and alpha proteobacteria. Furthermore, the number of losers much lower in invertebrates than in other eukaryotes and bacteria. Overall, on the first step, this result confirms our prediction. But what about ND6 gene? ND6, as a gene that's coded on the counter chain, should show a different result. So we can see that in vertebrates, the number of losers higher than the number of gainers. Then we took all eukaryotes mtDNA reference checks and repeated our analysis for more detailed taxa. In this box plot, taxa are located approximately in groups that make up the evolutionary series of mitochondria. The selection of individual taxa was based on the amount of available mtDNA in this taxon. We got the similar result at all. We can see the trend, but taxa selection and placement need improvement, and we will be grateful for a consultation on this question. The same trend and decreasing the number of losers we also can see for more detailed taxa, but an increasing losers among invertebrates is scary. Possibly, we can take a closer look on it when we build the correct taxonomy. The same results we had for ND6 gene, a decreasing number of gainers and a increasing number of losers. We explain all these trends by the mutational spectrum of mtDNA, and we have a few assumptions how it can be possible. Firstly, groups of organisms with a high rate of mtDNA point mutations have a greater shift in amino acid composition. We know that animals have a higher rate of mtDNA point mutations than plants. Therefore, an amino acid shift in animals should be greater than in plants. Secondly, the order of the taxa corresponds to the decreasing average effective population size. So, NE vertebrates are higher than invertebrates, than plants and fungi. This correlation can be interpreted as a long-term accumulation of slightly deleterious variants from losers to gainers in taxa with strong genetic drift, low NE. Our results are in line with the accumulation of the slightly deleterious variants in taxa with low NE. However, we can't rule out additional potential effect of the mutational rate, which is higher in animal versus plants, but other taxa-specific estimations are scarce. Asymmetry of the mutational bias, which must be stronger in vertebrates due to, for example, potentially more prevalent asynchronous mtDNA replication versus other taxa with variable modes of mtDNA replication, and global selection. Altogether, we suggest that the described abelian year trend of the amino acid substitutions in mtDNA of eukaryotes is driven by the asymmetrical mutational process. Thanks to all colleagues who works on this project and introduction to it. It's Alima Galieva, Alina Mikhailova, Viktor Shamansky, Kristina Ushakova, and head of our lab, Konstantin Papadin. See our website if you want more information about our lab. If you have questions or comments, you may send email to this address, and we will be grateful for your help. Thank you for your attention.